Welcome to the lecture on epidemiology. So in epidemiology, we're studying the ecology of disease. We're going to look at how relationships between a host and a disease occur, what these relationships are like, the different forms of them. If we can understand these types of relationships, then we can start to make some predictions and determinations. We can figure out when an outbreak might occur, be able to predict one. If we know that there's a certain species of mosquito in a certain area and its population is increasing, we can predict that there may be an outbreak of a disease that that particular mosquito might carry. We can determine risk and transmission patterns. We can say that so many people may be at risk within a certain 100-mile radius of that particular area in which those mosquitoes are increasing in population. We can look at transmission patterns. We know that it's going to start in one area and kind of build outwards, particularly looking at areas where mosquitoes may be breeding, standing water, those sorts of things. Understanding the ecology and relationship between a host and a disease is critical to developing treatments and different therapeutics, and most importantly, developing different ways that we can prevent these diseases from spreading. If we can prevent exposure to the vector, then we can thereby prevent disease. Some important terms or terminology in epidemiology. First of all, an outbreak is when there is a multiple number of cases in a small geographic region, such as we had an outbreak of Zika virus here in South Florida. An epidemic is when there are a large number of cases occur, and this usually happens in a very short period of time within a similar geographic region in terms of size. And we're going to be looking mainly at epizootic. Epizootic outbreaks occur. These are epidemics that occur in non-human hosts, and pandemics occur in human hosts. Pandemics is an epidemic that's just kind of gotten out of control. So the MERS virus started in South Africa, but that MERS virus has now that Middle Eastern, it's called the Middle Eastern Respiratory Syndrome, and it's caused by a virus. And that virus started in South Africa, over in Morocco, and eventually it spread throughout not only the African continent, but because of modern travel and how small the world really is, it spread pandemically or around the world quite quickly. It's usually considered a pandemic is a worldwide outbreak. This is an example of understanding the ecology of a disease. We know that this is Venezuelan equine encephalitis. It is a virus that is spread by mosquitoes, and we understand the life cycle of this virus in different organisms. So if you look at the top, we have a type of relationship called an enzootic relationship. And you can see between the mosquito and the rodent, there's this kind of rotating life cycle. The mosquito picks up the virus from the rodent and the mosquito transmits it back to the rodent. So as long as it's staying outside of the human realm, it's considered an enzootic disease. But mosquitoes are not necessarily always species specific. So female mosquitoes, they are looking for a blood meal. So if that blood meal happens to be a human after having taken a blood meal from a rodent, and that rodent was infected with VEE, then the human's going to be transmitted. In that case, when it moves from that animal to animal, that enzootic relationship, and it moves over to humans, it starts to become an epizootic relationship. So it can move from enzootic to epizootic. If we understand how these relationships occur, at the bottom there you can see with Venezuelan equine encephalitis that we get what's called an amplifying host. Now an amplifying host, the horse here in particular, it's considered an amplifying host because multiple other organisms were going to come into greater contact with it. So there's still research being done to determine if bats, who tend to live in rafters in the barns and such, may actually carry Venezuelan equine encephalitis, and they could actually transmit it to the horse, or they could pick it up from the horse. And the same thing with ticks. Horses do get ticks. The reason that those two particular organisms are of concern is because it's more difficult for a mosquito to actually bite a horse. Their hide is very, very thick. So they can only bite them around the ears and nose and eyes. and very soft tissue on the underbelly of these animals because the rest of their hide is too thick and the mosquito can't penetrate. But when that mosquito goes from a horse to a human, we're moving out of that enzootic relationship into an epizootic. Over here on the right, we have reported Lyme's disease cases here in the U.S. in 2001. And by mapping or modeling the transmission and movement of this disease and where this disease is occurring, we can see that it's highly concentrated over here on the East Coast, in the Northeast Coast in particular. And this is because the species of tick that transmits this disease is over here. And then we have some more cases over here up in Michigan and parts of Wisconsin. You can see there's spatterings of it across California, Oregon, Washington, Texas, down here in Florida. We have had a few outbreaks here in Florida, but nothing nearly as intense as what you see in that New England coast up there. So what are we talking about when we talk about a disease in a population? There is some more terminology that's important. An incidence is the number of new cases of a disease in a population in a defined time interval. So what this really means is how many new cases have been diagnosed in a specific population in the US or in Florida or in women or in children under the age of 10. So it's a very defined population, and it's also going to have a defined time interval. How many cases of Lyme's disease have occurred in Florida between 2002 and 2010? That incidence is going to tell us how many new cases. If somebody in 2002 already had Lyme's disease from a previous year, they would not be counted. So incidence is new cases. Prevalence is the total number of cases in a determined period of time. So in this case, in 2016, there were 134 confirmed cases of Lyme's disease. That does not necessarily mean that in 2016, 134 people contracted Lyme's disease. It just means that we know at that point in time in the Florida population, there are 134 cases. Some may have gotten it before 2016. When we discuss a disease as an endemic, this is a disease that's always around. It has a constant prevalence or a constant presence in a population. Malaria is endemic, even though it's a vector-borne disease. Common cold, the flu. These are diseases that we see all the time, and they're very common. An enzootic is endemic in animals, and if we have a zoonotic disease, this is a disease that non-vertebrates can transmit to humans. Anthraponosis is a disease that is human-specific. HIV is an anthraponotic disease. It only occurs in humans. Syphilis is actually an anthraponotic disease, and as is gonorrhea. So what are these different types of relationships that we were talking about? Well, we have mutualism, where both species of organism will benefit, such as our gut bacteria, skin organisms. We have commensalism, where one benefits and the other one is fairly unaffected. Skin flora is oftentimes considered a more commensal relationship. For example, not every single bacteria in our gut is a mutualistic organism. We also harbor some organisms in a commensal relationship. They just happen to be part of the population and they're there. They benefit by living in our intestines. They have nutrients and warmth and water, but they don't really do anything for us per se. When these organisms get transferred, that's known as foracy, and foracy is a part of disease transmission known as mechanical. In parasitism, one is going to benefit at the detriment of another. This is sometimes also referred to as predation. And in amensalism, the growth of one organism will prevent or inhibit the growth of another. So in amensalism, our gut bacteria are sometimes amensal for us, and they will prevent other bacteria from being able to grow. They'll occupy niches or outcompete for nutrients, those sorts of things. So that brings us to the idea of vector incrimination. In vector incrimination, this is a term used to talk about the role of an arthropod. How we're going to incriminate that particular, we're going to accuse a particular arthropod of harboring a disease. And so, how do we make sure that we've got the right vector? How do we ensure what criteria has to be met in order for a specific arthropod to be considered disease carrying? So how involved is the organism? We have to think about that, right? We have to think about a secondary vector. Sometimes there can be secondary transmission. They're not necessarily the primary form of spreading a disease, but they might pick it up and then move it. That's another form of foracy, mechanical transmission. Flies are oftentimes secondary vectors. The primary vector is the primary transmission route. In the case of malaria, that would be malaria and the mosquito. So there are 4 primary criteria that have to be met in order to accuse a particular arthropod or organism of being a vector. We have to first show that the arthropod commonly feeds on vertebrate hosts. And these hosts have to be hosts of the actual pathogen that causes the disease. So they either have to feed on the host or they have to make some type of contact with the host that would be effective in transmission of disease. Such as if you had Venezuelan equine encephalitis, just because a mosquito lands on you and it may not bite you. So that mosquito might land on you, but if it doesn't bite you, it's not going to pick up that disease and be able to spread it. So we have to show we know that these mosquitoes are landing on hosts, on disease hosts, specifically for a blood meal. We can observe that, we can see that, so we know that it meets that criteria. We have to show convincing biological association between the vector and what's known as a subclinical infection of the host. So if someone comes down with malaria, we have to be able to show that there was association between the mosquito actually biting the person and the diagnosis of malaria. So that one has to precede the other. We've got to show that there's some type of association between the vector and the host. So the host does not get infected until after being exposed by the vector. We have to show that vectors harbor the disease whether they are on a host or not. So we can't take somebody out in the wild who has malaria and get mosquitoes to bite them and then grab those mosquitoes and say, oh yeah, the mosquito has malaria. We have to be able to collect mosquitoes without a host and show that these mosquitoes in the wild on their own actually carry the pathogen. And we have to be able to determine and demonstrate the actual mechanism of transmission of the vector in some type of experimental condition. So we have to show that when a mosquito bites someone that they actually inject a virus or a plasmodium or a bacteria, whatever it may be. Whatever disease-causing agent, we have to be able to show that mechanism of transmission. How does the vector transmit the disease to the host? We have to not only be able to figure it out, but we have to be able to show it and show it in laboratory conditions. Another term, or 2 terms actually, that we need to look at are vectorial capacity versus vector competence. And these are very much the same but very different at the same time. So the relationship between a vector of an infectious disease agent and the actual host itself, those 2 relationships, have competence. How does or what makes that vector the correct organism for a particular pathogen? Again, I'm going to use the malaria example. Why are mosquitoes just the perfect vessel, the perfect vector for that malaria plasmodium? What is it that makes the mosquito good for it? Not only is it blood feeding, but the mosquito is a very important part of the sexually reproducing part of the life cycle of the plasmodium parasite. So it's important to be able to figure out the connection between the appropriateness of the vector and the particular pathogen that it is transmitting. How does the behavior of that vector or its physiology, or both behavior and physiology, contribute to transmission? Again, back to the mosquito. The mosquito is this kind of perfect vessel for this type for transmission. It is blood feeding, so it has direct access to the bloodstream of warm-blooded mammals, and it constantly is looking for it. It's attracted to CO2, which we exhale. It is blood sucking. It has changes in body temperature. So the Plasmodium gametes can mature in the mosquito gut, but don't mature in our bloodstream. So there's lots of little things that are kind of put together. So between its behavior, it's a blood-sucking insect or a blood-feeding insect, and its physiology, it doesn't maintain a high blood temperature. It doesn't have that homeostatic temperature. So with a cooler temperature, it allows for the gametes of the Plasmodium parasite to mature, and thereby sexual reproduction can take place within the mosquito. This in turn promotes genetic diversity of the Plasmodium organism, making it very difficult for us as secondary hosts to get rid of it or prevent it. Now, vectorial capacity. Vectorial capacity is used to determine what's known as an entomological inoculation rate. We're basically looking to see how well and how efficiently the vector can transmit the disease, right? So what's its capacity for disease? Its competence is how good is it at it. Its capacity is how much of it can it do. And this is sometimes related together, and they are somewhat interdependent on each other. But the vectorial capacity is a big calculation. Now, I'm not going to ask you to make this calculation, but I do want you to have a general understanding of the different components of it. So when we compare capacity to competence, in capacity, we look at the number of vectors that are available. We looked at how many of those vectors are actually going to bite. So we might have 1,000 vectors available in a 24-hour period, but only, if we have 1,000 available, maybe only 100 of them are going to actually bite or take a blood meal. What part of that population actually feeds on a specific reservoir? So maybe we might want to know, out of those 100 that bite, only 20 of them bit humans and the other 80 bit a rabbit or a goat or something else. How long do these vectors live? How much time occurs between their blood meals? If they're blood feeding, how much time occurs between them? And how long does it take that pathogen to actually incubate and carry out that part of the life cycle within the vector itself? When we look at vector competence is about the factors that influence the mechanics. So the three major components of competence are the uptake of the pathogen by the vector. Is it dependent on the ability of the vector to get the pathogen? Do they have the right mouth parts? Is the proboscis strong enough? Is it long enough? Do they inject an anticoagulant so there's no blood clotting if they're blood feeding? Are they biting? Or is it a solid? What's the actual mechanism occurring? What about development of the pathogen? This is where we look at that temperature difference in the the gut of the mosquito versus in the human bloodstream. The mosquitoes, they don't maintain temperature. They're like cold-blooded, so to speak. So those that blood meal will cool off, and if there are gametes in that blood meal, they will actually mature in the cooler temperatures of the mosquito gut. This is why the Plasmodium will sexually reproduce in the mosquito, but not in the human. And then what about transmission? Can the vector effectively deliver the pathogen? Can it take it from one host to a new host? Can it move it from one to the next? So if it's if you think about honeybees, when honeybees sting something, they lose their stinger and the honeybee dies. So that particular organism would not meet all three components of vector competence because it's not really able to transmit a pathogen effectively from one host to another. Mosquitoes, on the other hand, can take multiple blood meals from multiple hosts. So they're able to move that pathogen around from host to host. So this is a little blurry, but this is vectorial capacity in mosquitoes. And vectorial capacity is 9 times out of 10 being calculated for mosquitoes because it really depends on blood meals. So it's MA squared VBP sub N over negative log sub E times P. But what do all of these different things mean? This is all just a fancy calculation where they take some different data and different factors or components of vectorial capacity and created a formula to come up with a number. So MA squared is based on successful biting data. How many encounters with a host, were bed nets present or not, and did the host exhibit any kind of defensive behavior? And those three scores are given a value, and that's where we end up with our MA squared value. And VB, we get, is the mosquito itself immune to the host? What is the state of the midgut? So is it, does this particular mosquito have any kind of physiological difference in the chemicals or compounds found in the midgut? What are the parasite rhythms? So this would be things like fever cycles and the actual different developmental cycles of the parasite itself. In transmission, which is part of that VB value, in transmission competency, they're going to look, are there other organisms present in the midgut of the vector of the host? Does the host have an immune system that's effective against the parasite? And what's the quality of the host blood? P sub n, the n is what's known as the extrinsic incubation period. This is just basically the environmental temperature once the blood meal has occurred. And then P is the daily survival. So again, this is affected by host blood detoxification. How do they metabolize the blood meal? Does the mosquito have any kind of immune factors? Are there predators? Are there insecticides? And what's known as a desiccation risk, which would be a dehydration risk to the mosquito. These are all complex factors that are observed and measured. All of this data is then collected and put together to determine this vectorial capacity. So again, back to these components. Once these numbers are figured out, there's a couple of things that a vectorial capacity can be used to determine. How many individuals per unit of space? So the density in relation to the host. Are there 1,000 mosquitoes in this particular quadrant that we're looking at, or are there only 50 mosquitoes? That's going to make a difference, especially when you're trying to calculate something. Is the density of the mosquitoes affected by a population factor? If it's rainy season, we're probably going to see a greater population as opposed to a dry season. Is there competition? If we're looking at a particular species, then we can have competition between different species or two completely different genre of insect. Abundance is absolute abundance, the total number of individuals in a known population, and relative is numbers we use when sampling. So if we're not in the laboratory and we can't literally count exactly how many mosquitoes are present in a population, we can take some sample numbers and do a few calculations, and that can give us what's known as relative abundance, which is just a general idea of how many mosquitoes are in a given zone, a given area. And then we have that carrying capacity. And the carrying capacity is when a population of vector reaches the maximum capacity for a particular area. This isn't so much about the pathogen, but about the population. It's more of an ecological, more of an ecology type term. In carrying capacity, we have to take into consideration that a population is going to reach a maximum. We don't have enough food left. There's not enough blood meals left, or there's no area. There's nowhere else to lay eggs or have offspring. So an environment is going to be limiting. And there are going to be mortality factors in different environments, and how those factors affect different vectors can be measured and observed. So what we're really looking at is carrying capacity, because remember, we're looking at, in epidemiology, we're looking at that relationship between the environment and the vector and the host. So environment is going to play a really, really big role. We need to know the reproductive capacity of our population of vector. When we're going through this, think about mosquitoes. So in reproductive capacity, how capable is a population, how capable is it to increase in numbers, to get bigger, right? More people or more mosquitoes. And the ability of a population of an insect to increase is going to be directly affected by carrying capacity within that population. It's expressed mathematically as R sub 0 equals B minus D, where B equals the number of births observed and D equals the number of deaths. Now, in a group of animals like elk or maybe white-tailed deer or herd animals that are observed out in the wild, reproductive and carrying capacity is a lot easier to observe. But observing this in an animal or an organism such as mosquitoes, well, that's a little bit different. That can be a little more difficult. If the carrying capacity of the vector population is not reached, we're going to have an R sub 0 value. So if the R sub 0 value is greater than 0, then we know that the population is going to increase because we have not reached that reproductive capacity or carrying capacity. If R sub 0 equals 0, then the population is stable. We have an equal number of births and deaths. Think about the formula there, right? If B equals D, then R sub 0 is going to be 0. And then R sub 0 is less than 1, it's going to mean our population will decline. So if we have more births than deaths, right? If we have more births than deaths, then our population is going to increase. That makes sense. If births and deaths are equal, it's going to remain stable. And then if births are fewer than deaths, so deaths outnumber births, then population is going to decline. And when R sub 0 is less than 1, when the carrying capacity affects a population to the point that reproductive capacity is less than 1, is less than 0, is in the negative, then we're in concern. We are concerned for loss of that population. Now, I know we're talking about epidemiology and not ecology, and these are all more ecology terms. However, the ecology of the organism greatly affects the epidemiology, or the spread of disease, the behavior of a disease in a population. So in conservation ecology, we would definitely want either we would want our carrying capacity to be at least, in a perfect situation, it would be equal to 0, but we at least want it to be equal to 0, even greater than 0. But we would not want it to be less than 0. We don't want our population to decline in conservation ecology. In epidemiology, it's the opposite. So in epidemiology, we want the deaths to be greater than the births. We want to decrease the population of the vector because the vector is spreading disease. However, we don't necessarily always want it to reach 0. We don't want the population number to reach 0 because they probably do play some role in, as a producer or a consumer, or in the whole tangled web of life, but in one of the hierarchy levels. But we don't necessarily want the spread of disease. So we would want that to be a little bit different. Other components that affect a vectorial capacity in mosquitoes is going to be flight range. So how far can these guys move? Once they take a blood meal, how far can this female mosquito fly away? And that's going to depend on the range. What's interesting is I was watching a video the other day trying to figure out how they do it. There's an actual experimental machine you can purchase to tether a mosquito. You can tether a mosquito in the laboratory and measure their range. You can also, more commonly, they do what's the mark release recapture. It's what we used to do with honeybees. And in the mark release recapture, you capture you capture the vector, the insect, in one area, and you mark it with for the honeybees, we used to use different colors of nail polish. We would use something, a marker or polish or some kind of marker on the organism, allow it to fly, and then go to another area and hopefully recapture vectors that have been marked and see how far away they are from the original area. The A and the C equation in that vectorial capacity equation is based on the host's ability to bite. And it's affected by biting frequency. So what host does it prefer? Does it have a host preference at all? And if it does it feed only once? Does it feed multiple times? Does it feed once on one host and then again on another host? What is its habits when it's feeding? Then, of course, we have how long is this thing going to survive? So daily survival, they use a life table or calculate it. And vector competence, how well does the vector transmit disease? All of these are components of that vectorial capacity. How well, or how much can this organism actually hold in terms of disease. So here's just a little diagram that kind of throws together all of the different components in vector capacity and competence. There are extrinsic factors, so things that occur outside, environmental factors, temperature, nutrition, land use, rainfall, the environment. Then you have some intrinsic factors, which are usually physiological factors and physical factors of the vector itself. You have to also take into consider host immunity and genetics. For example, if you have sickle cell anemia, somebody with sickle cell anemia is going to be immune to malaria. They can't get malaria because the sporozoites can't get into their red blood cells. So that's host immunity. Some hosts are easier to penetrate or get blood meals off of than others, and mosquitoes are oftentimes attracted to one type of host more than they are another. And then, of course, how dense is the population at the time, and how much contact does the vector and host have together? So vector competence, how do we determine it? What are some of the things that are done? It's determined by looking for the pathogen in pools of hosts. So they'll collect a whole bunch of mosquitoes and run some kind of, we used to do this over in Dr. Kern's lab. We would go through the mosquitoes and we would find those that had recently taken blood meals. And then those blood-fed mosquitoes were then run through a series of tests for different viruses and diseases that may have been present. So we would use PCR for that. It's reported as, when they take these pools of vectors, they're going to report it as a minimum infection rate. So what's the ratio of disease to vector? And they're going to look for the presence of the pathogen. Most of the time, PCR nowadays is the most common method to do this, and they can test for multiple viruses in a whole host of different vectors. By looking for the pathogen in the host, this is known as a xenodiagnosis. They're going to use clean vectors to feed on suspected hosts. So let's say we have a rodent that's infected with a form of Plasmodium. We take a pool of mosquitoes that we know have not been exposed to malaria. They are not carrying the Plasmodium, and we expose them to the host itself. If the mosquito comes back after a blood meal and the mosquito is now infected, is now a carrier of the pathogen, then we know that the host was definitely infected. There are lots of host and pathogen factors that can affect vectorial capacity. Host immunity. In host immunity, this could include vaccinations. Vaccinations induce something known as herd immunity in a population. And of course, the existing immunity or existing immune health of individuals in different areas. I'm sure there's different levels of immunity in different parts of the world in terms of certain diseases. The susceptibility of the host is the presence of the disease what we call a silent host, or is the person asymptomatic. If someone is asymptomatic, it means they harbor the disease or they carry the particular pathogen. They're infected by the pathogen, but they have no signs or symptoms. Is there a high abundance of host available? Are we looking at a herd of cattle or just a couple of cattle? And what is the preferred host of the vector? Ultimately, the final thing that's looked at is characteristics of the pathogen itself. How virulent is it? Some diseases are worse than others. If it's a virus, it's going to be very, very specific about its host and what type of tissue it infects. But if it's a bacteria, it's not going to be nearly as specific. So that's something to keep in mind as well. Finally, we have the landscape epidemiology, and here in landscape, we're looking at the biogeographic relationship between a host and vector. So we're looking at a relationship between the host, the vector, and how the environment affects that. When we study the relationship between geographic features and the incidence of disease, so this would be extra rainfall during rainy season. Here in Florida, we have a rainy season. So weather and climate. A focus area if there's a high transmission risk. In our case, what if there's in Venezuela they have a really big problem with parts of Venezuela that are considered slums. And in those areas, there's a lot of standing water and stagnant water that can breed mosquitoes. So they get outbreaks of yellow fever, and dengue fever quite often. Weather and climate play a big role. Rainy seasons, dry seasons, they can both harbor different vectors and pathogens. What type of vegetation is present in the environment? If there's enough vegetation, proper vegetation, many insects are going to use that for laying eggs or having young. And then, of course, human culture and behavior. Are we using an insecticide? Are we wearing DEET or long clothing? Are we behaving in a way that's going to keep the mosquitoes away? Now, how do we assess risk? In epidemiology, one of the primary functions or primary jobs of an epidemiologist is to assess risk. And we use the behavior of the vector as a major component in predicting risk. We want to take a look and say, how at risk is a specific population. So right now, here they're looking at, this was, I believe, yellow fever. They were looking at the spread of yellow fever. And as we move through time, between mosquito eradication and other types of measures, we see less and less of this disease. So this is 2020, this year. So where do we see it? In the mosquito belt, more along the equatorial, more along the equatorial lines. You can see it down in here. This may actually be malaria. I forgot to put the note there. And then we have less and less in 5 years. Just in 5 years, they're predicting, and by 2040, the prediction is to have even fewer. Look, there's like just a tiny little spot right here. So there's a couple of different ways that we assess risk and predict movement or predict outbreaks of disease. One is through passive surveillance and the other is through active. In passive surveillance, this is usually done in the healthcare industry where they track reported cases. So healthcare workers, there are hundreds of diseases that by law a healthcare worker is required to report to the local health department. Malaria would be one, although we don't really have that here in the U.S. or South Florida. We do have yellow fever, which would be what we call a nationally notifiable disease. So that's passive surveillance. But the problem with passive surveillance for some diseases is there's a lot of underreporting. Underreporting is because a true case is not always defined well. So even healthcare workers may not know the true definition of a case. Some places just don't have the resources to do it. Other places don't have adequate medical care. So if there's not adequate medical care, people are not being seen or being diagnosed. And so there we have our lack of diagnostics. Another contributing factor in passive surveillance is the inability to diagnose some diseases until it may be too late for some organisms. In active surveillance, it's exactly that. We're just going out and looking for cases. We're sampling mosquitoes. Active surveillance is used more as a preventive measure. UF has a big active surveillance mosquito program. They're constantly surveilling mosquitoes out in the Everglades and testing them for different viruses and diseases to make sure that we know what is present out there, so that if there's a problem, we know where to look and we can prepare for that. So we start seeing an increase in certain viruses. One of the problems with active surveillance, though, is it depends on where you're going. Here in the U.S., you're with a research institution, you just get a permit and you do your research. But there are places that there are cultural barriers. You can't just walk into a forest in Peru and start sampling. Other areas, there are not enough resources. You would have to bring in a lot of resources, and that may be cost prohibitive. So there aren't a lot of options when it comes to active surveillance. In the disease reporting pathway, this is kind of an important one. We have healthcare providers, laboratories like UF over there, and other research institutions. All three of these, certain diseases have to be reported to the county or local health department. All sexually transmitted diseases and all mosquito-borne and all arboviruses, mosquito or vector-borne diseases all have to be reported to the local health department. and they have to be reported. I think it's within about 24 hours of diagnosis. There are a few other entities, some odd researchers, those sorts of things, and they will report these diseases directly to local health departments or the state health department. Ultimately, the Florida State Health Department, about once a month, will report all of its cases to the CDC. The CDC will maintain that for the entire United States, and the CDC also works with other countries. There's a couple of different new approaches to epidemiology. One, with the introduction of computers, there's been lots of new modeling and testing methods and those sorts of things. We have, of course, remote sensing, GIS, and surveillance programs. So, hey, that mark, capture, release, I can, or capture, mark, release, I can mark that mosquito and I have a GIS point, and then I can find that insect later on in another place and have another GIS point. So I have more accurate measurements. I can do that more quickly, more real time, and it gives me better data, more accurate data. In biochemical testing methods, there has been the introduction of what's known as a rapid test. And the rapid test is a way to just put blood. You may have done one at the office for, say, strep throat. They just take a small drop of blood, they put it on a filter paper, and it runs up the paper and tells them if the person has malaria or not. It's very, very similar, honestly, to a pregnancy test. Another method is PCR analysis, polymerase chain reaction analysis. That's used in the laboratories and hospital labs quite often, but it's very expensive and equipment intensive. So somewhere out in bush country, in the middle of Australia, you're not going to be able to run PCR, but you could maybe carry a rapid test for whatever disease it is you're looking for, if a rapid test exists. There are not rapid tests for every single disease out there. Another is genetic analysis. We look at isoenzyme studies, so what different enzymes are found and how are those evolving over time genetically. Another is the genetic flow of the vectors over time. How do these insects evolve, and is the presence of the disease or the pathogen part of that evolution pattern? This is what a rapid test looks like. This is a rapid test for malaria. And you can see here, don't forget, it always is going to have some kind of control. So this is the negative control. And that's what this red line is over here. Here, if there's nothing, then it's considered an invalid test because there's no control line. You can see here, C, that's a control line. And then there's the test line. So here we have P. falciparum, and here's P. vivax. These are plasmodia. And we can tell the difference between the two because falciparum has these two bands here, and Plasmodium vivax only has this one here. So not only can a rapid test definitely diagnose malaria, but it does it very well, very effectively. It can also tell us what species of mosquito may be present. This top line up here, you notice here it shows up in the negative result. That's because it is a control line. So if the positive control changes color but nothing else does, then you know that you have a negative result. So that's known as the positive control. And here's, you can see we've got all three bands there on P. falciparum. Other modern approaches include the evolution of parasitic relationships. What kind of relationships are occurring or changing over time between the parasites themselves and the host or vector. This is also done genetically through cladistics, so you can have cladograms and tables with percentages of relationship between different species and subspecies of organism. And then in epidemiology, they use a lot of modeling. This is mathematical modeling. It's done with numbers of cases and things like vectorial capacity and competence and those sorts of things. So all of those numbers get fed into computers, and they do a lot of different models. Now, if they do a stochastic model, this is to determine probabilities based on a set of random variables that were selected. And that can be different for whatever probability they're looking for. A deterministic model has no random variables. The data is very consistent, and it's just used to kind of determine presence of something. A heuristic model is going to combine a bunch of different mathematical solutions and it's going to combine them in a sequential manner to give us some records. So here's a stochastic model of Lyme's disease, and it's just a map giving us our risk factors. So stochastic models are oftentimes used to assess and kind of predict risk. And if we just put a bunch of numbers out there, then everybody would just look at us like we're crazy. They wouldn't know what we're talking about. But if we can interpret those mathematically, those numbers, we can manipulate them mathematically to show up on a map like this. You may have done something like this in a GIS course. GIS is great for modeling, and it's awesome skill to have for epidemiology. So anyway, this is Lyme's disease, and it's showing the different risk assessments. So that is it for our slideshow. I hope it wasn't too long for you. Exam 1 is this week, and if you have any questions, be sure to let me know. Have a great night.